We are starting the nervous system in anatomy. And by starting the nervous system, we are looking at the overall organization of the nervous system and looking at the nervous tissue. Now, the nervous tissue chapter is chapter 13 in your textbook. So, the nervous system is actually divided into two systems. Okay, so it includes all the neural tissue in your body, but it has two divisions. The first division is the central nervous system, or CNS, and that includes the brain and the spinal cord. The second division is the peripheral nervous system, and that includes all of the nerves that hang off of this brain and spinal cord, and they are called the cranial nerves and the spinal nerves, and they're sensory receptors that are attached to them. And they communicate with the central nervous system and peripheral tissues. Okay, so there's a communication between the peripheral tissues and the central nervous system. So these are the nerves that hang off of the cranium and the spinal cord, and they reach the far ends of your body. So all of those nerves hanging off, they reach the tips of your fingertips to the tips of your tippy toes, and all of your viscera, okay, and glands have these peripheral nerves. So that encompasses the nervous system, CNS and PNS. Okay, so the general organization of the nervous system. We said there were two divisions of the nervous system, so now we're looking at it in a more subdivided way. So look at this diagram. You see that we have the central nervous system on top and the peripheral nervous system towards the bottom, but we see a lot of subdivisions. We also see some arrows. We see the arrows are starting with the left-hand side on the bottom. We see that they go up. We see something called receptors, somatic sensory and visceral sensory, and then they go up. And they go up towards the peripheral nervous system, towards the central nervous system, and then we see information processing, and then it goes down towards motor commands, and then that goes all the way down towards effectors. So that's the direction of the organization. So let me explain. Okay, so in the nervous system, the organization is that you're going to take in, your body is going to take in information from receptors, from either somatic sensory receptors or visceral sensory receptors, taking information from the environment, okay, and then you're going to, that information is going to go towards the peripheral nervous system. Now, what does that mean? It means that the peripheral nervous system, remember, it's those either spinal or cranial nerves, or they're actually nerves that are taking in that information via receptors, moving it towards afferent, towards sensory information, towards afferent, towards the central nervous system, because the central nervous system is where we're going to take that information and process it. So your spinal cord and brain are going to process the information, and then it will say, well, what am I going to do with this? Okay, and when it decides what it's going to do with it, it's going to take that information and it's going to go back the other way, efferent, and there's going to be a motor command within that efferent division, and it will either go in one direction, the somatic nervous system, which is going to be your skeletal Your your somatic nervous system will include information for muscles, joints, skeletal muscles, joints, and the skin, or it will go towards your autonomic nervous system. When you think of autonomic, think of automatic. This is something that you don't think about, okay? And you can see parasympathetic, sympathetic. We're going to have a whole chapter on that. But these are things you don't think about. You don't think about your heart beating or glands secreting or smooth muscle contracting. Okay, so this is your viscera. Okay, so viscera. So that will be, and these are effectors. So the motor commands will make the effectors do their job, do that command. So this is just a big picture of the general organization of the nervous system, and it includes the central nervous system, the peripheral nervous system, but it gives you an idea of the direction of the commands, of how you receive sensory information, how it's two types, somatic and visceral, how sensory information includes receptors to receive the environmental stimuli, and there's an afferent division. So afferent means going towards with the sensory information. There's a processing area in the central nervous system. And then there's a motor command within the efferent division, going away from the central nervous system. And then the effector is what the command does. Okay, and it can be in the skeletal muscle or in the viscera, depending on what part of the peripheral nervous system you are at, if you're voluntary or involuntary. So we're going to get into that a little bit more, but I wanted you to see the big picture. So to reiterate, the nervous system has three major functions: sensory, monitors internal and external environment through the presence of receptors, okay, taking in information via the afferent pathway, integration up there in the central nervous system, spinal cord, brain, okay, integration of sensory information, processing, this is complex higher order functions, and motor, efferent, okay, response to information processed through stimulation of effectors. Okay, stimulation of effectors, and that could be a muscle contraction or it could be glandular secretion. Okay, so we're looking at it in words. So let's take a look at the histology of neural tissue. There are two types of neural cells in the nervous system. There are the neurons and what's called the neuroglia. The neurons are the functional cells of the nervous system. They are used for processing, transfer, and the storage of information. We've seen neurons before in our study of histology, and we'll go over their structure in a little bit. Neuroglia, or glia, are for support, regulation, and the protection of the neurons. And they actually outnumber the neurons. Okay, so you can use the word glia instead of neuroglia. Now, glia, there are actually separate glia for the central nervous system and for the peripheral nervous system. And we'll look at that shortly. So the functional cell is the neuron, and neuroglia are for support, regulation, and protection of the neurons. And the neuroglia are going to actually outnumber the neurons. And I've seen different numbers for this. I know there are at least 50 to 1. So there are 50 neuroglia to one neuron. But I've actually seen different numbers here, so um, it's controversial. Different texts have different numbers, so they way outnumber the neurons. The neuroglia way outnumber the neurons. And let's go further and move on. So here are the neuroglia. Remember, I told you that there are different glial cells for the peripheral nervous system and for the central nervous system. So I guess the way this diagram is situated, it starts with the peripheral nervous system. So we'll go with that. Now the satellite cell is the first cell that we're looking at on this diagram. Now the satellite cell is a cell that basically it surrounds the neural cell bodies in the ganglia. What is a ganglia? A ganglia is a term that we use only with respect to the peripheral nervous system, and it basically is it is a term used for cell bodies in the peripheral nervous system. It's like a bump, an oval bump in on a sensory neuron. You'll see it, and it includes cell bodies. So the satellite cell is going to regulate chemicals that are around these cell bodies in the ganglia. Things like oxygen, carbon dioxide, neurotransmitters, the levels. So it monitors those levels around the neurons in the ganglia, the cell bodies. That's the satellite cell. Okay, next one, the Schwann cell. There's only two, remember? Now the Schwann cell is pretty special. The Schwann cell, and we're going to go more into detail of each of these glial cells. But the Schwann cell is going to do two things. Now, if you look at that axon there, because they're not showing you the whole neuron, but the Schwann cell is going to myelinate the axon. And what is myelin? Myelin is basically composed of lipids. Okay, it's composed of lipids, so it's going to wrap around that axon, wrap around. And the reason for it is, we're going to tell you about that afterwards too, but it's going to form, when we have a myelinated axon, it's going to leave little nodes in between each myelinated section of the axon, and it's going to allow for faster transmission of an action potential. So the innermost part of the neuron on the axon is going to be myelinated. But after that, if you look more superficially, we're going to see that the Schwann cells does something else. It's going to make an additional sheath, and that's going to be called the neurolemma. Okay, and that neurolemma is going to allow the peripheral neuron to do something that the central nervous system neurons cannot do, and that is it's going to allow them to regenerate. Okay, as long as the cell body is intact, the neurolemma will allow the peripheral nervous system neurons to regenerate. Okay, so moving to the right side, again, we're going to go into detail about these glial cells. So we have the central nervous system glia, and we'll start with the oligodendrocyte. Now, notice the difference between the oligodendrocyte and the Schwann cell. This guy can myelinate. It's a myelination glial cell. It myelinates the axons of central nervous system axons, but it can do it. It can myelinate more than one axon at a time. Okay, remember the Schwann cell myelinated one axon at a time. And it does not form a neural lemma. Okay, so that's the main difference. Okay, oligodendrocyte. Then we're going to have the astrocyte. So the astrocyte kind of looks like a star. Okay, and they have little like feet attached. Okay, and it's a cell, so it has a nucleus, of course. Okay, it's going to help maintain what's called the BBB or blood-brain barrier, provides structural support, regulates, and this is also going to be similar. We can, this is kind of a cohort to the satellite cell. So we can liken it to the satellite cell in the peripheral nervous system in that it regulates nutrient and dissolves ion concentrations, just like the satellite cell. Okay, and the other thing it does is that it's going to form scar tissue after injury. Okay, then we also have the microglia. Now you can also pronounce this microglia. All right, so you can pronounce it both ways. This is the immune system cell or macrophage of the central nervous system, and it removes cell debris, wastes, and pathogens by phagocytosis. Okay, gobbles them up by phagocytosis. We all know what that is. Okay, but let's not forget the ependymal cell. That's how you pronounce that. And these are, and you're going to see another picture of this. These are lining cells. These are going to line the ventricles of the brain. And you don't know what those are yet. We're going to learn about those. These are not a typical lining cell. You know, the typical epithelial lining cells are simple squamous. These are simple cuboidal, and then they have little processes here. They have cilia, usually. Microvilli are cilia. And they're going to assist in producing what's called cerebral spinal fluid. Okay, we're going to learn more about that. So these are the 6 cells called neuroglia or glia. Okay, let's move on. Okay, let's do this slide here. This slide is showing you a picture of what the central nervous system and its glia look like. But first I want to give you a little bit of an introduction to something else. If you look at the top of the slide on the far right corner there on the top, it says white matter and gray matter. So I know that you probably have heard this a lot. And so I just want to clarify it for you. So that is a cross section of the spinal cord. And you can see the gray matter is that horn looking thing there that we're going to teach you about. And then you see the white matter around it. So what does that mean? Okay, just so you probably have been wondering about that. So gray matter, actually what that means is that it's the soma, the cell bodies. Okay, it means mostly cell bodies. Okay, so and then the neurons come out and the axons come out. You know, cell bodies stay in that horn and the axons come out and they're myelinated. So myelination is made of lipid and it's white. It's whitish because it is lipid. And so we call that white matter. Okay, so it's mostly axons coming out and the gray matter is mostly cell bodies. All right. So now let's focus our attention on the left-hand side and that pretty image there. Whoever drew that was very nice. Did a good job. So let's look at the top. We're going to talk about oligodendrocytes and microglia in here. So I guess I'll skip that ependymal cells for now. So let's look at the oligodendrocytes. The oligodendrocytes, if you can find them, these guys are pretty neat. What a neat glial cell this is. He's got his arms around 2 axons. 2 axons. So what he's doing is he's myelinating 2 axons at one time. Okay, he can do more than one axon at one time. He's wrapping around. Now, not all axons are myelinated, but a lot of them are. And he's wrapping around that lipid material around the axon. Okay, and more than one at a time. Okay. Okay, so what is the purpose of the axon? It actually allows an action potential to move down the axon more rapidly. Okay, because what happens is action potential, and I'm not going to go into the physiology, but it jumps across the myelinated um, material to the internode. If you notice that there's a node in between. If you see the word node, that's a node, and it jumps over the myelin to the node, so it actually creates faster transmission. Okay, that's all I'm going to tell you. All right, so that's the oligodendrocyte in the CNS. The other thing I'm going to talk about in this slide is we'll look at the microglial cell right there, and that is your brain macrophage. That's your immune cell. We've seen them every place, haven't we? Okay, we've got to have those guys. So they phagocytize cellular waste and pathogens. If there's cellular debris, if there's bacteria, anything that's going to get in the way, this little guy is going to undergo phagocytosis and gobble them up. Okay, so we'll leave it at that for now, and we're going to look at another one of these pretty pictures again. Haha, I told you we would find another nice illustration of the glial cells in the central nervous system. And this time we're going to focus on the astrocytes. So the astrocytes are the star-shaped looking cells, and hence the name astrocyte. So focus on the astrocyte. Okay, and there's a couple of them there. So if you want to maybe look at the lower one that actually has the label astrocyte, that might help. And the astrocyte has little feet. Okay, they stick. They're sticking to that axon there. Okay, and they will stick to blood vessels. Okay, and they have a big cell body and a nucleus. And they are also sticking to the basement membrane of a capillary. And that's going to be very important to one of their functions. Okay, so what are the functions of astrocytes? Okay, they create supportive framework for neurons, but what this is doing here right now on the blood vessel is it's creating the blood-brain barrier, or BBB. Okay, so it's not alone in this. It facilitates the blood-brain barrier, but it's those little feet they're closely knitted, and they when big molecules want to get through and get into that get into the brain, and we don't want them there, the astrocytes will not allow them. So that's what the blood-brain barrier is about, not letting certain things into the brain. Okay. They also are very similar, the astrocytes are, we can liken them to the satellite cells, as they monitor and regulate interstitial fluids around surrounding neurons, like CO2 and oxygen and neurotransmitters. So in that way, they monitor the levels of those things getting into neurons, cell bodies. Okay. They secrete chemicals for embryological neuron formations. And an interesting thing they do is they stimulate the formation of a scar tissue secondary to central nervous system injury. So they do quite a bit, astrocytes. Okay, so the next glial cell we're going to talk about is also of the central nervous system, and it's called the ependymal cell. Ependymal cell. Okay, so these cells They're pink. They're a lining cell of the central canal, and they are, if you take a look on top there, they're pink, and they're simple cuboidal cells with processes. See them up there? So they line, and their processes are actually going to, they line the ventricles of the brain and central canal of the spinal cord also. So they're not only in the brain, they're in the central nervous system, and the central nervous system, remember, is the brain and the spinal cord. So we need to remember that. And both of these places are going to have what's called cerebral spinal fluid. And these cells are the cells that secrete them. So they produce this cerebral spinal fluid. Also, you can also call it, abbreviated CSF. So they produce, monitor, and help circulate CSF, cerebral spinal fluid. Okay, so there they are. So they're aligning right there. Okay, ependymal cells. Ependymal cells. So we've gone through the glial cells of the central nervous system. Now it's time to go and talk about the two glial cells of the peripheral nervous system. And the first one we're going to talk about is the Schwann cell. Okay, so the Schwann cell is the myelinating cell of the peripheral nervous system. So we need to look at this a little differently, because remember, the Schwann cell only myelinates one axon at a time. Okay? And there's going to be two things that are created here. So if you look at the axon, okay, so you see the cell body or soma, and then you see the axon. And it's going to have two parts. So the innermost part covering the axon is going to be, there's going to be myelin covering it. Now, myelin is made of lipid material, and it's wrapped around several times. Okay, so that's covered. But in addition to myelin, okay, the Schwann cell also is going to create something called the neurilemma. Okay, a neurilemma is an additional material that is not found in the central nervous system. So if you take a look at that image there, you see the neurilemma on the outside. Okay, it's an extra sheath. Okay, it surrounds all axons of neurons in the PNS. Okay, and what it's going to do is it's going to allow regeneration of damaged axons. Okay, as long as as long as the neuron is not, as long as the soma is not damaged. Okay, so that's kind of special, because this neuron can regenerate because of the neurilemma. Okay, so one axon at a time, and it's going to be myelinated, okay, right around the axon, and then there's an extra sheath called the neurilemma. And remember that the myelin sheath is going to allow for faster axonal transport. The second cell that we find, the second glial cell, is called the satellite cell. And this cell, you can liken it to the astrocyte of the central nervous system. It doesn't have as many roles, but the one role that it does have is that it's going to support the ganglia of the PNS, and it's going to monitor chemicals, oxygen, CO2, neurotransmitter, the levels that go into and out of the cell bodies of the ganglia. Okay, so there you are. but we're never quite finished talking about these things. So let's go over them again. And the central nervous system glial cells, we have the astrocytes that regulate the microenvironment of neurons, also help create the blood-brain barrier, also known as the BBB. They serve for structural support. They repair damaged nervous system, known as scar, and they're involved in neuron development. The oligodendrocytes, they myelinate the central nervous system axons, and they can myelinate more than one axon at a time. Okay, little note here that not all nerves in the central nervous system are myelinated. I'm going to show you some of the neurons, and I'm going to show you one that's unmyelinated and one that is myelinated. Okay, we have a lot of different types of neurons, and I'm also going to tell you about those too. But some of them are myelinated and some of them are not myelinated. Microglia. Okay, these are the macrophages of the CNS. They get rid of debris and pathogens. Phagocytosis is the most common way. Ependymal cells. Okay, these are linings and they are composed of simple cuboidal epithelium, and they have processes and they produce cerebral spinal fluid, CFS. And we'll go over the PNS glial cells. We have the Schwann cells, myelination, and they also produce the neurolemma. All peripheral nerve axons are myelinated. So there you are. All peripheral nerve axons are myelinated. White matter. Nodes of Ranvier, you know what those are, right? In between the myelination is the node. So you skip over that myelination and you jump to a node of Ranvier, named after the guy called Ranvier. Okay, the neurolemma. Okay, that allows those neurons in the peripheral nervous system to regenerate. Okay, it allows the neurons to regenerate. It's the superficial sheath around the myelin and axon. Okay, so it's above the myelin. Satellite cells, okay, these are actually monitoring chemicals that go in and out of the ganglion cell bodies. So finally we get to the neuron. Now, this neuron is an unmyelinated neuron, okay? So many neurons are myelinated. Just as we said, all neurons in the peripheral nervous system have axons that are myelinated. So if you look at this axon, there is no myelin there. But let's talk about the parts of the neuron. Okay, so don't worry. I know there's a lot of terms here, but we'll get through it. Okay, so if we look from the left to the right, we see on the left-hand side, we see a big cell body. This is called the soma, and it's covered in this type of a neuron. This is actually called the multipolar neuron. There are different types of neurons, and we'll get to that. So the soma is covered with dendrites, so they look like little tree branches, and as they branch off, their extensions have little pointy things, and those pointy things are called dendritic spines. Okay, so we have the dendrites and the dendritic spines, okay, for incoming information. Now let's go into the soma, and we see, like all cells, or most cells, can't say all, we have a nucleus, and inside the nucleus we have the nucleolus. Okay, but we have a special name for the for the nuclei. One is nucleus of neurons, and we call it a perikaryon. Okay, so I put that up there on the top in red. Okay, it just has a special name. And then we have the chromatophilic substance, and this is also called Nissl cells. Okay, so and what it does, Nissl bodies, what they do is it stains. Okay, so it stains purple. Okay, so it has a pigmentation effect. Okay, in neurons. Okay, if we start moving down towards the axon, we're going to see a little piece of segment called the axon helix, excuse me. And that is a place that is going to, where we're going to stimulate what's called the axon potential. So we don't learn about that in this class, but that's where it happens. Okay, it either happens or it doesn't happen. It's an all or nothing effect. Okay, so it's called the axon helix. Okay, and it's right over there. Okay, and then we have the axon. Okay, did I not talk about the Golgi apparatus? We have Golgi apparatus in the soma there too. Okay, so moving down the axon, which maybe may or may not be myelated, and then we go to the end, which is the axon terminal. But at the axon terminal, it segments into terminal boutons. And we've seen this in another chapter when we talked about the synapse. And the synapse we talked about was the synapse with a neuron and the muscle, the neuromuscular junction. So the terminal bouton is, I have it circled right there at the axon terminal, right at the end of the neuron. So it ends, and then what it will do is it will synapse with a postsynaptic cell, which could be another neuron or a gland or a muscle cell. So we'll talk about that separately. But this is a neuron, and the movement is going to travel from the cell body to the axon terminal. That's the way the movement will go. So I thought I would show you a myelinated neuron. Okay, because I showed you an unmyelinated neuron. So here's one with myelination. Okay, and I also said on the last slide that the direction of the impulse went from the soma or cell body to the axon terminal, and then to the boutons, terminal boutons. So it's the direction of the impulse. Okay, so there's the myelination, and then you can also see that there are little gaps in the myelination, and those are called nodes of Ranvier. Okay, so this just gives you a picture of a myelinated neuron. And because I like to say things over and over again and show you slides again and again, but in a different way, I'm going to show you the neuron structure again. So here is an unmyelinated neuron, and I'm showing you it in parts. I'm showing you the dendrites, which are stimulated by the environment, take in information, but this is just anatomy. There's your spines, and then we move over to the cell body or soma, which contain the mitochondria, ribosomes, and other organelles, inclusions, cell bodies, remember that, perikaryon, remember that. And then the conduction will move forward towards the axon. Form is for function, and it's long, so that's why we have that. Okay, the action potential towards the synaptic terminals. Okay, and here they are, the axon terminal, call it actually, and then you have the terminal boutons. Okay, which is going to synapse with another neuron or a muscle or a gland. Neuron structure. So these are the different types of, or the structural classification of neurons. They're classified based on their number of processes coming off the cell body. And I'm going to go into each one separately on future slides. So the first one's called an anaxonic neuron. All right, so the anaxonic neuron, it looks like it only has dendrites. Actually, when you look at this neuron, you just can't differentiate between the dendrites and the axon. Okay, it does have an axon, but we don't know which is which. We don't know which process is which process. So we call it the anaxonic neuron, and I'll go into more detail on a future slide. Okay, the next one is called the bipolar neuron. So the bipolar neuron actually has an axon and a dendrite. Okay, so it doesn't have two axons, it has an axon and a dendrite. Okay, so the next one is called the pseudo-unipolar neuron. I think this is my favorite one. So they call it pseudo-unipolar or unipolar neuron. Now, it's called pseudo-unipolar sometimes because it looks like this. If you look to the right here, it looks like this soma or cell body doesn't have a little process sticking out of it, or it's just so short that they call it pseudo. So it has that little process, but it's there, it's sticking out. And then it branches off into two axons. So it has two axons branching off of that little process from the soma. And it says two axons, and then it has dendrites on one side, and then it has synaptic terminals on the other side. And we'll go into the details on the next couple of slides. Right? And the last but not least, actually the most common type of neuron is called the multipolar neuron, and there actually are several types of this neuron, where we have the soma or cell body, and then we have many branching dendrites off of the soma, and then emanating from the soma or cell body is the axon leading towards the synaptic terminal. Okay, and then we're going to go into each one separately. This is the axonic neuron. So when I first introduced it to you, I told you that this neuron looks as if it doesn't have any axon. It looks like it's all dendrites, but in actuality, we just can't discern the axon. So we don't know. Okay, so as the slide says, no anatomical clues to determine axons from dendrites. So we don't know, which is which. Okay, this is what it looks like, and that's why it is named the anaxonic neuron. Okay, so there are really axons there. Okay, so we don't really have a lot of information on this neuron, but we know that there are some in the special senses. In the retina, we have the amacrine cell that's there. There are some interneurons. One is the amacrine cell, and we haven't learned about interneurons yet. You will. So this is what they look like. You just can't tell the difference. You can't discern axons from dendrites. They all look like dendrites, but this is the anaxonic neuron. Okay, moving on. Okay, this is the bipolar neuron. So check it out. You see the cell body in the middle, and you see the axon, and you see the dendrites on one side, and you see the synaptic terminals on the other side. Bipolar. So two processes coming off the cell body, one dendrite and one axon. That's what it looks like. So we call it bipolar. These are found in the eye, ear, and nose. And typically when we show you a picture of this, you'll see these in the retina. Okay, so the retina of the eye, you'll see the bipolar neurons. Okay. Okay, this is the unipolar or pseudo-unipolar neuron. Remember, I told you the reason it was called pseudo-unipolar is because that little process from the soma on the side there is just so short. So it actually has two axons, but one axon is leading to the dendrites and one axon is leading to the synaptic terminals. So there, it has direction. Okay, so we have two axons and synaptic terminals. Now these the single process coming off the cell body gives rise to dendrites at one end and making up and an axon in the other. Okay, making up the rest of the process. So you see that there. This is the pseudo-unipolar neuron or unipolar neuron. You can call it either one. And we find these often in the sensory neurons. So you'll see this in the afferent pathway. Okay, as you're going up the peripheral nervous system, you're going to see these in sensory neurons, and you're going to see that on the dorsal root ganglia when we talk about that. Okay. This is the most common type of neuron. This is the neuron that I show you when I talk about what is a neuron, what is the structure of a neuron. And we see the soma covered with dendrites, the axon, and the synaptic terminals. Okay, so multiple dendrites and a single axon and the synaptic terminal. However, I should tell you that there are many types of multipolar neurons, and I only have two examples here. I have the motor neuron, and I have something called the Purkinje neuron that's found in the cerebellum, just to show you that there's more than one type. Okay, and they all have those multiple dendrites and single axons. I could have given you a lot of examples, but I'm only showing you two. So the multipolar neuron is the most common type of neuron. Speaking of neurons and their functional classification, we'll give you a little bit of function, just going to define it. You should know that there are three types of functional neurons. We can classify neurons by function. The sensory or afferent neuron going towards the central nervous system. Okay, they transmit sensory information from receptors of the PNS towards the CNS. Okay, and those are, we're going to talk about them as being pseudo-unipolar neurons. Okay, most of them are unipolar or pseudo-unipolar. A few are bipolar. So remember, unipolar. And the dorsal root ganglia, we're going to take a look at that in our next presentation. The motor neuron, efferent, transmit motor information from the central nervous system to effectors. Effector is a muscle, gland, or adipose tissue in the periphery of the body. Okay, so remember the motor neuron is a multipolar neuron. And then we have something called the association or interneuron. So we haven't looked at that a lot. Okay, but they are going to transmit information between neurons in the central nervous system. They're going to communicate between the sensory neuron and the motor neuron. Okay, in processing. Okay, analyze inputs, coordinate inputs are the most common type of neuron, 12 billion. Okay, and they're going to be multipolar neuron. Should I put that there? So getting back to classification, the sensory neurons are part of the afferent afferent division of the peripheral nervous system, the somatic sensory neurons. They deliver information to the central nervous system, so they take in environmental cues, environmental stimuli, and this is somatic. So we're talking about your position in the world, joint position, tension in muscles, state of homeostasis, skeletal muscles, joints, skin, and this is mostly pseudo-unipolar neurons. Okay, these somatosensory neurons are pseudo-unipolar, and they're going to have the dorsal root ganglia outside of the central nervous system, okay, where the special sensory neurons, okay, are going to take in light, sound, and their neurons are going to be a little bit different. They're going to be bipolar neurons. Okay, think about the raw. If you we took a look at the picture of the retina rods and cones. We see bipolar neurons there. Those are called your special senses, or your senses that take in light and sound. So we have some bipolar neurons there. But those are also sensory neurons, but they're special sensory neurons. So the interneuron. Interneurons are the connecting neurons within the CNS. They're located in the CNS. And the example I'm going to give you is when sensory neurons are moving up the afferent pathway, the interneurons are going to be in the CNS, and they're going to connect to motor neurons. They're going to analyze these interneurons are going to analyze the sensory inputs. What do we do? So they're part of the processing. And so they're connecting the sensory to the motor neuron. They're coordinating the motor outputs. They're processing, integrating information. So they're in between. They're classified as either excitatory or inhibitory. So you don't know what that means yet, but just know that they're excitatory or inhibitory. And so I'm going to show you a diagram, and you'll get it in a second. So the interneurons, and it's just as it says, an interneuron. They're usually multipolar neurons. So let me show you what this looks like. And here we are. So we're moving up the afferent pathway and the somatosensory pathway. And there's your sensory neuron. Look at that, the pseudo-unipolar neuron. Okay, so envision this in the peripheral nervous system. Okay, you see your receptor endings there. Okay, you see your myelinated axon. You see your dorsal root ganglia. Okay, and then the dendrites, and then this yellow neuron here, multipolar neuron, is an interneuron. Is going to be in the central nervous system. It could be found in the spinal cord. So it's connecting, it's synapsing on the sensory neuron right there. Okay, so it's going to be processing the information from the sensory neuron. Taking that information, what do we do with this information? Okay, and then it's going to move on to the dendrites, synapsing on the dendrites of the motor neuron. And a motor command is going to move down the other way in the efferent direction and elicit a response to the effector. Now the effector is not shown here, but it should be found right after the axon terminal here. So it's going to be after the axon terminal. Okay, the axon terminal can be connected to a muscle, a skeletal muscle, or this is a motor neuron, so we know that it's a somatic. Okay, this is somatic tissue. So it would be a skeletal muscle, or it will be a tendon, or the skin. If you're pulling your hand away from a flame, a hot flame, or you touched a sharp object. Okay, so this gives you an idea of what the interneuron does. Okay, it's right there in yellow. Now let's look at those sensory receptors that are attached to the sensory neurons that are afferent or afferent. Okay, we can classify them in different ways. We can classify them by structure, but I'm going to classify them by location. Okay, now one of the sensory receptors is going to be located outside the body, and that's going to be called an exteroceptor. Okay, exteroreceptor. Okay, exteroreceptor. And these are basically found if you touch your body, the back of your head, okay, and they sense, okay, the environment through touch, temperature, pressure, okay, in your skin, and also your special senses. We take in the environment in vision, hearing, olfaction. So these are all called our special senses, and we have special sensory receptors for these special senses. So these are called all part of this division of exteroreceptors. Okay, the other two receptors are found inside the body. I'm going to go to the bottom one first. Now, on this slide, it's written as interoreceptor, but in your book, it is written as interoceptor. So I guess both ways are correct. So, but as I'm saying, interoceptor is what is written in your book. Okay, so this is an internal sensory receptor, and it monitors the internal environment to maintain homeostasis, which is balance. So what does that mean? Okay, so basically it's going to monitor the digestive, respiratory, cardiovascular, urinary, and reproductive systems and provide sensations of deep pressure and pain, as well as taste and other special senses. So we said that special senses were exteroreceptors or exteroceptors, but some of them are interoceptors as well. So we have two types, okay, for the special sensors. It just depends what they are. Okay, so taste is an interoceptor. Okay, so these are maintaining the internal environment. Okay, all right, the other internal sensory receptor is called the proprioceptor. Now, there's only one way to say that, okay? Proprioceptor, okay? The proprioceptors are, they're in the skeletal muscles and joints are involved with this receptor, okay? They monitor your position and movement and your basic position in space, okay? So your position of your joints and your skeletal muscle in space. Those are proprioceptors. And when you get to physiology, you'll see how that really works. Okay, so these are so inside the body, proprioceptors, interoceptors inside the body, and exteroceptors outside the body. Location. The structure of a typical synapse, and there are different types of synapses for this presentation. I'm going to show you the typical synapse. We've looked at it before. We have the end of the neuron, so the axon going down to part of the axon terminal, which is the boutron, terminal boutron, and we can call that a synaptic knob. And then we see that the knob is synapsing, so that's what a synapse is. It's basically it's next to, we have a presynaptic membrane of the first neuron, and then it's going to be next to either a neuron or another type of cell so that it can release its neurotransmitter, which is in a vesicle, into the synaptic cleft. Okay, and then it will be captured by the postsynaptic membrane of the next cell, which could be a neuron, or it could be a muscle cell, or it could be a gland. I'll show you that. So this is a typical synapse. Okay. So as I said, we many times will have a neuron synapsing with a neuron. So we would call the first neuron a presynaptic neuron, the second neuron postsynaptic neuron, just like this. And there are variations on this. Okay, so a second type of a synapse. So that would be a synapse. Second type of synapse would be a neuromuscular synapse. We've seen this one before, where the presynaptic neuron synapses on skeletal muscle. Okay, the third type of synapse that we're going to talk about today is the neuroglandular synapse. There, the neuron is synapsing on glandular tissue. Okay, a couple of cells, all the same. That's glandular tissue. So, and we know that what do neurons do? They're going to secrete neurotransmitter. It's going to stimulate, okay, something, the gland to secrete its glandular secretion. So, but we don't go into that because that's physiology, but that's the idea. Since this is the introduction to nervous tissue, and you're getting an idea of which way neurons go as we are, as you are stimulated by the environment, they go towards the central nervous system. They're processed, and then they move the other way with their process. There's an integration, and they move back the other way with command, and they stimulate an effector to do that command. So sometimes because this is the introduction, we don't see that those neurons are not alone, that they move in the nervous system. They tend to move in groups or bundles, and they are moving in bundles or nerves. So we want you to know that neurons, axons of neurons, are bundled together to form nerves. And we're going to show you this, and when we go to the next, we're going to introduce this to you now, very introductory. And then in the second presentation, you'll see it'll be much more apparent, and we're going to really go into detail of the anatomy, that nerves form tracks or pathways in the CNS. Okay, so remember we said that axons are myelinated, and that myelination forms the white matter in the cell bodies of the gray matters. But we're going to introduce what's called tracks or pathways. And we want you to know we want you to distinguish between a neuron and a nerve. And I'm going to show you that in a few slides. Okay, so it's kind of like we can remember the muscle and the muscle fibers and how that was all lumped together and big and the connective tissue. So we're going to look at that. So the cell bodies of neurons are clustered together. Cell bodies, remember, clustered together into ganglia. That word, ganglia, in the PNS. And the nuclei centers are in the CNS. So we don't call nuclei centers in the CNS ganglia. We only use the word ganglia in the PNS. We call them nuclei centers in the CNS. Okay, so ganglia. And I'm so old that I remember textbooks that used to call nuclei centers ganglia. And then we used to say, oh, that's wrong, you can't use that word. I'm serious. But ganglia is reserved for the PNS. These are unmyelinated structures that will be part of the gray matter. So remember, gray matter is unmyelinated structures, and cell bodies are unmyelinated structures. Gray matter. Now you know the difference between gray matter and white matter. So we're trying to give you the big picture, and you're getting to know the CNS and the PNS. And we're we're going to give you more information. I'm going to give you more information. The textbook's going to give you more information. So let's move on. Okay, let's give you one last classification of neurons, or actually we're showing you the entire nervous system and how information flows. And we're distinguishing the receptors in the nervous system. So we start with the receptors because as the somatic sensory nervous system goes towards the central nervous system, right? So we broke it up by location. So we have the interoceptors, exteroceptors, and proprioceptors. So we said that interoceptors are going to receive information inside the body, but these receptors are of the visceral type: cardiac, respiratory, kidney, respiratory. So they're going to sense internal stimuli, and of course, they go towards the central nervous system. Okay, and they're going. It's an afferent direction. So we start with the peripheral nervous system. You see the red line going towards the central nervous system there. And when that happens, they're going to come into, these neurons are going to synapse, we know that word now, with interneurons. And interneurons in the central nervous system are going to be processors. They're integrating information, processing. What do we do with this information? Okay. However, that red line goes down further to the visceral motor neurons in the central nervous system, because this is viscera. And the visceral motor neurons are going to elicit a motor response. But if you take a look, we introduce something different here. We show you that those motor neurons are of two types. We're going to have two neurons that synapse on each other. We have a preganglionic and a postganglionic neuron. We have two. And that's important to know. When we study the autonomic nervous system, you're going to learn all about that. And so once it synapses on that, they're going to elicit a response on the effectors. And we don't break it up into the parasympathetic and the sympathetic nervous system, but you know that this is here, okay? And these effectors are smooth muscle, glands, cardiac muscle, and adipose tissues, because this is an involuntary or autonomic nervous system. Okay, so then moving back up, we see that we have exteroceptors, and proprioceptors. One is an external receptor, which is the exteroceptors, and one is an internal receptor, which is a proprioceptor. Okay, so they are going to go afferent. Remember, the proprioceptor is found in muscles and joints. Okay, and they they take in information in your muscle spindles and your joints, and they regulate your position in space. Exteroceptors are found in your skin, your bone, and your muscles and joints also, but they're external receptors. Okay, so we have internal and external, and they're going to go towards the afferent. Okay, in the afferent direction again. Okay, so peripheral nervous system to the central nervous system, and we have to have an interneuron there also to integrate, process the information. But this time we're going to use a somatic motor neuron. After we go to the interneuron, we synapse with the interneuron, and then that synapses with a motor neuron, a somatic motor neuron. This is voluntary most of the time. There's actually an involuntary somatic motor neuron that I didn't tell you about. Okay, so but most of the time it is voluntary. Okay, and then when we get out of the central nervous system, this motor neuron is going to elicit a response in the efferent direction to skeletal muscle. Okay, and joints, and it is going to move that skeletal muscle. The skeletal muscle, an example, will contract. So say you touched a hot flame, okay, or touch something hot in your external receptor there, and so the response will be to move that, contract the muscle away from the hot stove. Okay, so this is just showing you the big picture direction that we started in the beginning, but this time you know a little bit more about the types of receptors, the meaning of the words, and the parts of the sensory neurons and the motor neurons and the parts of the peripheral nervous system, the sensory and the central nervous system, and what an interneuron is and what a synapse is. And hopefully that will be our conclusion for today. And so we can move on and talk about the peripheral nervous system next, and the parts of the spinal cord, and that's the central nervous system, and that'll be really a lot of fun. Okay, so this is the end. Stay tuned for the next chapter for more neuroanatomy. See you next time.